Hello, my name is Jeannette Guerra, and as Jason said, I was the author of Vining Davis Fellow in the Department of Entomology. And the Department of Entomology is the study of insects, if you guys didn't know. And I was actually interning in the Burke Lab. And so the Burke Lab focuses on research of wasps and flies. And so I was the only intern there. So I got to collaborate with other lab members, for example, Hannah Boomgarden and also Kelsey Kaufman. And so I also got to live in the dorms, and my dorm was actually 5 minutes away from the lab, so I didn't have to walk that far because some of the students actually take buses to go to class, which is very different from here where you could walk to class for 5 minutes. So that was actually really good that I didn't have to walk that far. And I also heard about this fellowship through my advisor, Marty. When I asked her that I wanted lab experience, she was like, oh, well, I could refer you to Dr. Gillenberg. And, oops, I went too far. And so these are the species that I studied. So it was Anastropha suspensa flies and the wasps. And so in my regular work, we had a quarantine room, and I worked in the quarantine room, and it had 3 chambers to get in. And in the room, there's this huge room with hundreds of flies and hundreds of wasps, and you can see the cages up there. And they're located in the same room, but on separate sides. And the 3 chambers are built so none of the flies or wasps escape, in particular the flies, because the flies are actually pests to citrus fruits. So if the flies escape, we kind of have an invasion, and we don't want that. So I had to make sure to close all the doors before you enter, and make sure to, when you leave, to close all the doors. So it was a process getting in there. And so you're probably wondering, well, why should we study flies, or why should we study wasps? So they're very important because if you don't want larvae in your fruit, then you should probably research on wasps and flies. And so these wasps actually are able to kill the flies by injecting one of their eggs and venom during, usually when they're in the fruit. And you can see a picture there of the wasp attacking the larvae, or the fly baby in more general terms. And so some of the flies actually survived the attack. So we were just wondering why some survive and some do not. And to test this, we have to inject the flies with beads to test their immune system. So that was the project that we focused on. And so I had a lot of different methods that I had to do. So I had a lot of injecting and dissecting of wasps and flies. So I had to inject larvae, and you can see the picture there. They're really tiny, and I have to inject them with glass needles. And I have to be really careful because the glass needles break easily. I broke about 10 when I was first starting, so I learned the process after. And also for the wasps, you have to take them out of the cage and then put them in this container and then put them in ice so they won't move while you dissect them. And then once you dissect them, you have to go under a microscope and then take out the venom and then throw away the wasp. So it was a long process. And for the microscope, it has a built-in camera, so it has to be connected to a computer. So you have all these things in a tiny little desk, so you had to be really careful. So those are some of the methods that I had to do. And so for the results, they were inconclusive because each fly had a different response to the injection. And but we did take all the pictures, which you could see up there. So one of the things that we concluded was that maybe we injected the flies at different spots, and that resulted in the variability in the results. And so what did I learn? So this internship helped me experience lab techniques. For example, I got to use a microscope that had a camera, which I hadn't done before. And so I also had to inject a lot of things, so I am very good at that now. But I also have to keep a lab notebook, and I had to keep a lab notebook before in classes, but it was not as intense as here. where you have to write everything down, and once you get results, you have to print the pictures and put them in the lab notebook for future reference. I also got an understanding of what it is to do research, and I had to repeat a lot of my experiments often. So I had to be very patient, and that was one of the things that I learned. And so this internship helped me build connections between other scientists and other research, and we also got to cooperate down the lab with Strand's lab, and so I met more different people. And Dr. Galenberg actually mentioned that if I want to do graduate research in her lab, I was welcome to. So that was an opportunity that I had. But I mentioned that I wanted to join Peace Corps before I do graduate school, so I made sure she knew that. And these are some of the pictures around town that I took. And so I just want to thank the Arthur Davis Foundation, my site advisor, Dr. Galen Burke, for hosting me, and also my faculty sponsor, Marty Condon, for giving me the opportunity to go to the lab. Thank you.